Karen Lloyd, thanks so much for coming on the podcast.
Happy to be here.
So, Karen, you're the Wrigley Chair in Environmental Studies and Professor of Earth Sciences at the University of Southern California, a job I think you moved to after 12 years as a professor of microbiology at the University of Tennessee. And for today, you were the author of a book that came out earlier this year called Intraterrestrials. So let's start with the title. What are intraterrestrials?
It's organisms that we only discovered like a few decades ago. And there's so many of them, and they're so weird that they're kind of like extraterrestrials. It's kind of like aliens, but inside Earth. So the name, that word intraterrestrials, is meant to be an inverse to extraterrestrials.
It's a great term. And yeah, so it's essentially, is it fair to say they are subsurface microbes?
Yeah. Yeah, for the most part. And I also include sort of surface microbes that are being supported almost entirely by subsurface stuff. That's just because this is where we are, that's where we tend to like get some samples. And I use an inclusive way of defining it, I think.
Right. So one concept we're taught in school is photosynthesis, which is how plants basically convert sunlight or convert energy from the sun into glucose and power themselves. And then, of course, we eat plants, or we eat animals that eat plants, and we survive off of that converted sunlight. For microbes beneath the surface, there's no sunlight. So how do they power themselves?
They use chemical energy instead. And when you use chemical energy to power yourself, which we do, we use chemical energy to power ourselves, but we don't make biomass from it. We're not autotrophs like plants are. But these things many of them are autotrophs. They're like plants, but instead of sunlight, they're using chemical energy. And that opens them up to a whole big range of possibilities that are so much more varied than just interesting chemically than what happens up at the surface.
And so the way this happens, right? So if it's a microbe that's essentially right next to some sort of rock underground, if that rock is made of a certain chemical element, if that microbe might be able to basically feed off of that element from the rock, right?
Yeah, right. And what they're doing is they're respiring it. So it means that they respire a rock. So they breathe rocks, which is, it's funny to think about because, then it like feels like putting rocks inside your lungs, but of course they don't have lungs. And breathing in its essence is just moving electrons onto a chemical that's good at taking them up. And they can do that with a solid mineral.
Yeah. So in terms of, to get some other technical terms out of the way, what is serpentinization? I may have mispronounced that, but serpentinization, and why you write that it may be even more important to life than the sun is. Why is that?
Yeah, because possibly, if we didn't have serpentinization, we wouldn't have life. There's some theories that serpentinization is a reaction between a set of rocks and water. And that's it. That's all you need to make it go. The simplicity of that chemical equation has been sort of tantalizing to early Earth, origins of life researchers, because it produces chemicals that are more oxidized and also ones that are more reduced. And so it sets things up so that you can have respiration, you can get chemical energy from things, and you can basically drive life from serpentinization pretty much on its own without needing anything else.
You mentioned earlier that these microbes they respire other elements than oxygen. We breathe oxygen, so we need oxygen to survive. We think of that sort of as the norm on Earth. Even when we talk about kind of living on other planets, one of the topics is always, is there enough oxygen there? To what degree might we actually be the exceptions on Earth? Or we're sort of the newcomers as far as what we need to breathe?
Well, we know that we're the newcomers because there wasn't a lot of oxygen on Earth until about 2.4 billion years ago. There's debates about whether there was a little bit of oxygen before then, but it certainly wasn't very abundant until then. And so there's a whole, at least 1.4 billion years of life being here where there was not oxygen. So definitely a lot of that early evolution of life happened without a lot of free oxygen around. So for that reason, we know we're not the dominant ones on the planet. But also, just if you look at the real estate of Earth, if you extend where you think the habitable zone is down into the Earth's crust, then you realize there's just a lot of places you can live if oxygen is not something you're dependent on.
Right, right. And in some of these places, they're pretty extreme environments. Could be really low pH or really high pH. Could be extremely high temperatures. There are microbes that have adapted to live in really low pH environments. Could be liquid that if we touched ourselves, our skin would burn off. But they can live
Yep.
there.
Yep.
How are they able to do this?
They are like amazing little machines. They have a bunch of ways of keeping the protons out. So that's what acid is. It's a bunch of protons, a bunch of positively charged hydrogen atoms. And so they build their cell walls kind of like tanks. They're basically less porous than a lot of other organisms, which presents challenges because a single cell needs to be somewhat porous to get food in and to get waste out. So the fact that they have to be so controlled about it is a problem that they have to solve. And then another thing that they do is that they fight the positive charges by just building up positive charges that are kind of inert and not so dangerous on the inside. So they'll fill up with positively charged potassium, for instance, just to like counteract that waterfall of positive charges that are basically just constantly trying to get inside. And then the last ditch effort, which they have to do all the time, is that they just pump it out all the time. It's just like bailing out of a boat. And they're constantly doing this. And this means that if that is your lifestyle as a microbe, then you need to have an energy source that is commensurate, that is enough. You're bringing in enough energy to be able to afford all this stuff you have to do to deal with the acid.
So we know how they can survive in low pH environments. We also we know that they can survive in high pH environments, but we don't exactly know why, right?
Yeah, I don't think I think there's been a lot more research in the acid than there has in the bleach. I mean, people have done work, and it's basically a similar type of thing. They build themselves like tanks, and there the problem is that your protons are going to leave. They're going to just fall out because the liquid around you is so devoid of any positive charges, of any protons, that the ones that you have inside you, which you need to sort of make your cell function, if there's a hole in your membrane, they're just going to fall out. And this presents a weird challenge because one of the basic things that we know that life does is that it pumps protons across the membrane. Mm-hmm. And that's creates this thing called the proton motive force. And that gradient that we set up, and we're doing this right now in all of our cells, mm-hmm. That gradient that we set up it's what fuels the production of our ATP. It's what fuels our ability to get energy when those protons rush back in. The thing about the high pH organisms is that if they did that, if they tried to set up some kind of gradient like that, they could never do it, because the medium around them is so devoid of any of these protons that they can't keep them. They can't make that store be a big reservoir that's going to come rushing back in, because if they pump one out, it's just going to leave. So one of the things that they do is that they run their pumps off of something other than protons. Mm-hmm. So they'll do a sodium pump, which is really, those are quite common in life as well. But to run your whole cell off just a sodium pump is pretty weird.
Right. And is it fair to say that is their way of doing it is probably more sort of closer to how life was when it first appeared on Earth, and then we have sort of evolved beyond that?
Yeah, maybe. It's hard to say. You can look at like how closely related things are to deeper branches on the tree of life. And, they all go back pretty far. So life really radiated quite early on this Earth. But I don't know, it could be primordial. I tend to think like if life had evolved at pH 0 or pH 10, then our internal biomolecules would function at pKa's of those values. We would have that be the pH that determines our functioning. So that's not the case. Basically, most of these cells are circumneutral inside. Mm. And so I think that implies that circumneutrality was sort of where we got all our biomolecules set up.
Since we just touched on it, one of the implications of this body of research about these subsurface microbes is answering the sort of big question of where, how, and when did life start on Earth. And one of the leading theories has been that life started in these undersea hydrothermal vents, Where do you come out on that after a career studying subsurface
Yeah.
life?
I love the idea that life started in these vents, because I can just imagine what it was like in the late 70s for people who were studying, geologists were studying these temperature anomalies at the bottom of the ocean, and they had no idea that they were going to find the life that they did down there. I mean, they truly thought it was just heat and metals that were coming from these vents. And they didn't know until they took a submersible down there. And it just must have been, it must have been like landing on Mars and finding little Martians walking around. I mean, they got down there, and I've been to hydrothermal vents. They are teeming with life. They're crawling with critters, and they just had no. So that probably really fueled imaginations that this must be the origin of all life, if they can exist here. And I would never argue hard against that possibility. I think that is still, like, we kind of know now. The way I think about why hydrothermal vents are such a compelling place and they have so much biomass is that it's all about gradients. It's all about differences. And there's a huge difference between oxygenated, cold seawater and metal-rich 600 degrees Celsius water coming out of these vents. And those gradients make so many possibilities for a powerful reaction. So if that is an incubator of life, well, we know that it is in modern times, then the subsurface, I think, I want to say clearly, I don't know how life evolved on Earth. I don't know about the origin of life, and I would never want to argue hard any one thing. But I think the fact that there are so many gradients of chemical differences, like low-powered chemical differences, sort of calm, easy, womb-like even chemical gradients that could be exploited in the Earth's subsurface. I think it's something that we should think about, that we are essentially a subsurface phenomenon, and then we radiate it out. Once we got a foothold and we evolved some stuff and we got some leverage in the world, then we sort of emerged into the sunlight, the harsh, terrible sunlight, and we're able to actually take advantage of the great stuff the sun provides while also protecting ourselves from it.
Just to make sure I'm understanding, are you saying so the one of the sort of supporting ideas behind these hydrothermal vents being the area where life first appeared is that there are these sort of extreme gradients across a couple different dimensions? And if that's the sort of supporting premise of that theory, there are even more extreme gradients under the hydrothermal vents, essentially.
Yeah, I wouldn't necessarily say more extreme. I mean, there could be, but there's just more of them.
Okay.
They're not point sources. They're just kind of everywhere, because the subsurface is heterogeneous. You have different types of rock. You have rock veins. You have places that are sort of sucking in seawater underneath sedimentation in a situation where you have continents, which we do on modern Earth. You have sediments coming down, that doesn't need life to make it go. There's just a lot of possibilities for how to get this energy. There was another thing that was fueling those original ideas about hydrothermal vents, and that's that the organisms that people were finding that were deeply branching on the tree of life tended to have DNA that was like hardier. It was GC rich for people who know what that means, but it's basically just that they had more of the pairings that have 3 hydrogen bonds between them rather than 2. And so the idea was that our primordial life was for high temperature. But now that we have a lot more genomes on the tree of life, that is no longer the case.
So that's a topic you mentioned in the book. How impactful has DNA sequencing been to this entire field of research?
It's the whole thing. Like, we wouldn't know and that actually is, I think, a very interesting thing to think about, that we could, if we did not have DNA sequencing, if Carrie Mullis and the other folks involved did not figure out how to make polymerase chain reaction, that amplifies DNA, if they hadn't figured out how to really make that go, we would still be completely ignorant about this whole world. I mean, we would have cultures, we would know, you can culture things, and you can culture a lot of things. It gives you a really great view of the diversity of life, but we'd never know about the existence of these organisms. So it makes me think about, what are we missing now?
So where is DNA sequencing? Although it's obviously it's come a really long way. Where are there still some limitations in the sense that there are things you're particularly excited about once we can sort of clear those engineering hurdles?
Yeah. So one of the ways that we sequence right now, just the technology, is that we have to sequence in 150 base pair fragments. And a genome is like usually millions of base pairs. And so we take these little, we take our intact DNA that we pull from a hydrothermal vent or something, and then we chemically or physically chop it up into little bits. We destroy our own sample in order to sequence it. And then we get the sequences back, and then we do really fun, interesting computer programming or computer algorithms to figure out how they go back together again. So that's a fun puzzle, but wouldn't it be nice to not have to solve a puzzle at all and just look at the stuff directly? And we're moving in that direction. There's Oxford Nanopore Technologies is a
Yeah.
company that's got long read sequencing. It's not like quite taken over just yet, but I think it will probably soon. I don't know, as far as I can tell.
Hmm. When we take bacteria and we culture them in the lab, we're making them grow. We're essentially having the cells replicate.
Mhm.
But it's artificial growth. Like we're
Mhm.
modifying their environment to induce growth, maybe more than what would happen in nature. And it turns out that it might actually be a lot more than what is happening in nature. So what do some subsurface microbes teach us about growth and lifespans and the power needed to replicate?
So, one of the things that we can do in sediments is that we can figure out the respiration rates of an organism without growing them in lab, just because the sediments are layered very nicely. And we can do models that can tell us how quickly they're breathing. And we can do that calculation, figure out the number, and then we look at the number and we're like, holy crap, this is 10,000 times less power than you would need to just sit on a lab bench and do nothing.
Hmm.
Like people have measured the maintenance energy, like how much power energy over time it takes to keep just like a senescent dead, not dead, but not growing
Yeah.
population of microbes to just sit and maintain themselves. And what we're seeing is that on a per cell basis, it's like 4 or 5 orders of magnitude less than that. So the implication, and this is an implication, this is, well, we do actually have some ways to measure this directly, but I couch it a little bit just because it has to remain a hypothesis because it's really hard to prove this. But the implication is that these individual cells last for 100,000 years,
Right.
a million years.
What would the evolutionary reason for those that long of a lifespan be for those cells?
So that is a fun thing to think about. And I spend a lot of time thinking about it. You have to first consider that maybe there is no evolutionary driver for it. It just happens on accident. I do know colleagues who sort of assumed this or implicitly assumed this or argued this. And I think that that has to remain on the table. I think it's very unlikely. If you starve me, I'm not going to last for more than, a month max. Because I'm tuned to need to eat every single day. But I just don't think you can take like a normal E. coli population and just like have them last a million years if you stop feeding them.
Right.
So the other argument that there's special adaptations is that the things that we tend to see in the subsurface, we don't tend to see elsewhere. So they're like, it's their special place. Like they like being there. It's their niche. And so that would argue that like, this is a niche. This isn't an accident. Okay. So then if we agree with the premise that they are evolutionarily adapted to be there, then what are they waiting for? How does this even work? What's your payoff? And I like to think about it like seasons because nobody is confused about why a tree does not have leaves and does not do much in the winter because it's cold. There's not a lot of sunlight. It's waiting for summer. And we know it's waiting for summer because we see it happen every year.
Mhm.
So what are these organisms waiting for that they would get to then pass any mutation that they had that made them better at hanging around and doing nothing for a million years along? And to answer that question, we have to look at geology. We have to look at things that happen over longer time scales than seasons. We have to look at ice ages. So they could be waiting for their tectonic plate to crash into another tectonic plate and they get shoved up to the surface or something.
Right. Yeah. It's definitely sort of an interesting topic to think about because those events happen on such long timelines. It does make sense that maybe they evolved to be able to wait for those events to happen. There was another example in the book you talked about where I think I might butcher it a little bit, but basically cells that had evolved to essentially almost have the near stagnant growth would actually outcompete those that grow faster in a sort of like head-to-head competition for resources, right?
Yeah, this is called the growth advantage in stationary phase. And this is developed by Steve Finkel, who's one of my colleagues here at USC, is sort of the person who pushed this idea the most over the years. And what he does is that he takes literally E. coli, so the lab rat, something that I have never seen in the deep subsurface. So this is not like one of the professionals, but it does pretty well considering. And he starves it. And he'll starve it for years. And what he sees is that it doesn't just stop growing. He sees subpopulations that live and die, that are like competing with each other. And at the end of, this starvation, if he starves them for long enough, then if he takes a young culture that has not gone through this starvation and he mixes it together with one of these old cultures, and then he starves the system, the old guys just kill the young ones. Like they're good at starvation. They get good at it. And so it really argues that there is this phenomenon. If you study microbiology, you learn about lag phase, log phase, and stationary phase. And that's the growth curve that a microbe goes through. And we know that they do this in pure culture. And then maybe you talk about the death phase or the senescent phase, but it's kind of like, well, who wants to think about that? That's really dull. But if you look at where microbes are in the world, not just in the subsurface, but even in the surface, they're all in this senescent death phase. Like for the most part, they're not undergoing this like rapid log, everybody's dividing every 30 minutes situation. I mean, the exception being like when you start to get sick, like if you get strep throat or something, then they're going through log phase growth. But for the most part, they're in this death phase. And so even for the fast guys, even for the surface organisms, stagnation and death seem to be an advantage for them.
Is this a sort of a correct, probably overly simple example? So if a subsurface microbe that is kind of almost in a state of stagnant growth for 100,000 years, then a volcano eruption happens, you get pushed up to the surface, and then you're competing against the surface microbes, the ones who don't rely as much on sort of being in this sort of dormant state. You're actually going to, if there's limited resources, you're going to do a better job of winning those resources than the surface microbes.
Maybe. I don't think like in a head-to-head competition with an E. coli, I think that pretty much anything in the subsurface would never against an E. coli. But it's more just that we do see them coexisting with the fast guys. So it's more like they win by just not competing. The idea is that they are functioning on such slow timescales, and they're functioning in a system that doesn't wash them out of it. Like we don't see these things in our guts. I mean, you've never seen some of these deep subsurface clades in our guts, because if you don't regrow after, every defecation, like you're out. So you have to grow fast to live in a gut. But in places where you don't get washed out of the system, then the fast guys can be doing their competition thing, and you can be dividing, passing your genes along, evolving, adapting, and producing progeny that have the adaptations that allowed you to get there slowly but fast for your species, while these other species are burning out and they're not going to make it.
It's fascinating.
It's just a hypothesis too. I'm just working on it.
Yeah. So we talked about how DNA sequencing really kind of helped open up this area of research. I wonder, obviously, like the field work to study these microbes is pretty daunting, and you talk about you include a number of examples of the field work you've done in the book, and it's pretty gnarly. For lack of a better technical term, like it is pretty. These are like pretty. People would do these just for kind of the pure sake of adventure, but for you, it's work. Do you think that element has also maybe? It's been maybe a little bit why we've been sort of delayed in understanding this part of the biosphere.
It could be, it's and there's plenty of people who are involved in this research who don't even go to the field. So I kind of want to say that, because like I wouldn't want somebody who was interested and excited about, the computer programming part of this or the chemistry part of this or the lab part of this and think like, oh, well, I could never go in that direction because I don't want to, carry around a rifle for polar bears. But you don't have to do that stuff. I just like that part of it. So that's the end of it that I like being on. But there's plenty of people who don't. I don't know. I don't know if, we as humans are pretty drawn to exploration. So, but we don't get to do it that often. So that could be. I mean, that's definitely true for the deep sea. It is so remote and it's so hard to explain how hard it is to work in the ocean. I know that's like a silly, stupid, simplistic statement, but it is so hard to work. Everything we do, just walking up to a rock and looking at it, like you can't do that in the ocean. And yes, you can be in a submersible, you can do that, but you get your 9 hours, but actually the weather gets crummy up at the surface and they call down from top lab and they say, you got to screw up the dive, like right in the middle of what you're doing. So it's not, I think there's just like a ton of mysteries in the ocean for sure.
What's it like being in one of those submersibles? I mean, is it just pure adrenaline the whole time?
Yeah, for me it is. It's almost like too much to handle because you don't want to be. I feel like there's a sort of adrenaline induction that Hollywood tries to do, or media or like fictionalized accounts of exploration tend to hype the situations more than they are when you're actually in them. The act of like getting us in a submersible, closing yourself in and dive and like just descending down into the water is calm. No one's yelling. No one's acting like a cowboy. If somebody started acting hyped, I would leave the situation. I would be like, this is not a safe situation. But actually what it is a checklist, a literal paper checklist with a clipboard and like going through all the checks. I mean, it's kind of like flying. If you look at your pilots when you fly commercially, every single flight, they get out of the cockpit, even when it's cold outside, and they do a physical walk around the whole plane and they have a checklist. And it's the same thing when we dive in a submarine. It's very, very calm. But, internally, it's very hyped.
Yeah. Looks. I mean, you talked about how you saw some animals down there essentially that you didn't expect to see. There was 3 to 4 foot long worms and stuff like that, right?
Yeah, they can be 6 feet long. These are crazy. It's Riftia pachyptila. This is one of the things that grows around hydrothermal vents that it's an organism that we never would have invented in our brains if we hadn't seen it from a submersible. But it is gigantic white-bodied worms. And with these big red feather heads, and they are so evolutionarily tied to that hydrothermal vent that they don't eat themselves. They don't have mouths, and they don't. Normal worms, they pull in dirt, like an earthworm. It pulls in dirt through its mouth and puts it goes through its gut, goes out its anus. And all of those parts of a worm have completely gotten lost in these ones. And all they have is a sack of microbes inside them. And the microbes do the chemical reactions that provide food for them.
Mm-hmm. Yeah, it's fascinating. And I think you said when you were down there in the sub, if I remember correctly, you weren't expecting to see those there. It was kind of like, whoa, what are these things?
Yeah, well, I don't study them. So I'm there for the fluids coming out. I'm there for the subsurface guys. And so with the animals, and I always feel this way with animals, again, I do not study animals. I'm as much a tourist as anybody when it comes to an animal. I'm just like, what is that? I don't know. It's crazy.
Another area you went to for field work is the Arctic Circle. I think you went to a place called Svalbard.
Yeah, I was there 2 weeks ago.
Oh, really? Oh, interesting.
Yeah.
Wow.
Yeah.
And so were you there, because in the book you talk about you've been there to study the melting permafrost, right? Was that the same for your recent trip?
Yeah, so this is basically like we needed to go back and get some samples because we had an idea of an experiment that we wanted to do. And it's hard when you work in remote places. You can't just get an interesting idea and immediately test it in lab. You got to okay, is this idea worth it? Can we get funding? How do we get up there? The logistics and everything. And so this was an idea that we were like, okay, it's worth going, and we were able to get some funding to go. And so we tried this, but it is an experimental manipulation. That's just what we were doing this time, to try to figure out what's happening with these soils that are thawing, and they get mobilized and they move into the fjords around them. It's just glacial melt, either seasonal or climate change related, dumps just massive quantities of these soils into the fjords.
Mhm.
And so it's an interesting question, like what happens to them when they get there?
So how worried should we be about this? And there may not be a sort of clear, definitive answer at this point, but I guess what are the difference, the sort of arguments for and against us being super worried about it?
If you live or work in the Arctic, there's no question in your head. You see the glaciers receding. You see that you used to be able to drive over the ice in the fjord all winter long, and now you can't. It is so rapid. The Svalbard is warming faster than the rest of the Arctic, just simply because they get the Gulf Stream. They get the tail end of the Gulf Stream. So it's, even though it's like one of the farthest north pieces of land on Earth, it's 79 degrees north. It's warmer than everything else at that latitude. The answer is yeah, we should be really, really worried. I don't know what to say about it though. I'm certainly not like in any kind of position to like snap my fingers and pull CO2 out of the atmosphere. But
Oh, sorry, just to clarify, I didn't mean the warming and the melting. I meant more sort of the exposure of these microbes that have been living in the permafrost.
like the
And
pathogens.
right, exactly. Because
Oh,
you talk about
okay.
how on one hand, there could be a lot of elements that are released into the air that have been buried for a very long time. On the
Yeah.
other hand, it's possible these microbes might actually help us pull CO2 out of the air because that's what they do underground anyway,
Yeah.
or in the permafrost. So yeah, is that with that context in mind? Is that sort of what you've been there to study?
Yeah. And so, one thing that is a little hopeful that we're seeing is that it does look like the methane, which, pound for pound ends up being more impactful for warming the earth than the CO2, tends to be cleaned up within the soils. So we see that the upper layers of soil are using it and the lower layers of soil are making it. So that is one kind of positive aspect, is that they may be sort of mopping up some of their own greenhouse gases, which is really nice.
Yeah. Yeah, it's interesting. Awesome, Karen. I think we've sort of motored through my questions. Is there anything I haven't asked that you would want to convey?
I think that, like, I guess it depends on who I'm speaking to, but I think that, like, just the notion that, like, I'm not an anomaly. Like, I when I started doing this stuff, like speaking publicly about my work and stuff, I was so worried about being seen as a soul, because we have this like idea that scientists are these like lone wolves sitting in a laboratory, like making great discoveries and thinking great thoughts, and we set them apart. We set them as like these geniuses who function on their own and give them a Nobel Prize. But in my experience, the really good way to move science forward is to talk to each other and to work as a group, and think your great thoughts, be stand out, all that kind of stuff, but like to function all together. And so that's the message I always want to send to people, is like, science welcomes you. We need a lot more people. We need a lot more scientists, and we need scientists who come from different perspectives than the people who normally do science. We need the people who normally do science, like, professor kids who grew up in that world and know that world very well. We need those
Mhm.
people, but then we need people who wouldn't even consider themselves to be associated with science and would never think of themselves as being a scientist, because that diversity of thought and diversity of perspective is I think where real innovation comes forth. And I just think that that's something that we really need. And basic research, research that doesn't have a return on investment. You can't see where the profit's going to come from. The only thing that's driving it is that you don't know the answer. That's it. That's where big discoveries come from, is blue skies research. And those two things are important.
You did end the book talking about the importance of teamwork, and you had some examples of from the field work you've done, and I didn't really sort of interpret it in that way that sort of trying to say science is not just sort of kind of sitting in a lab doing semi-isolated work. It's about the team of all these different folks who sort of move our scientific understanding forward. Awesome. Karen Lloyd, thank you so much for coming on the podcast. Thank you for writing this book. Page Turner, I learned a ton. Highly recommend it for folks out there. And yeah, thanks again.
Thanks so much. See you later.
Cool.